Um, Last talk of the morning, Dr. Cassandra Quave is going to talk about PhytoTEK, a company that she put together here in Little Rock before moving to Emory. She's back today to talk about it. Cassie, you're on.
Okay. Well, thank you all for having me here. It's a real pleasure to be back in Little Rock today. So, let me see if I can find here we go. So I'm going to discuss our company PhytoTEK. I'm the CEO and co-founder of PhytoTEK. And basically what PhytoTEK is we are an infection control company. So we are looking at both treating and preventing staphylococcal infections. Here's a little bit of an overview of what we'll discuss today. First of all, to highlight the point that staph infection is a very important and expensive problem across several different industries. Our team at PhytoTEK combines both scientific and business experience, and I'll go a little bit more into the management in a little bit. Our product is a plant-derived natural product composition that specifically targets biofilm formation and is useful in preventing recurrence of infection. And I'll explain what a biofilm is, too, in a moment. We are pursuing a slice of a $3 billion market opportunity, and we're doing this in a very strategic way. As you all know, pharma industry is a kind of a risk-filled industry for small startups. So we're trying to mitigate this risk by going after small market opportunities in the veterinary space in particular, and then moving our way up into the human market. Our core business model can generate between $200 to $300 million in revenue, and today we're seeking somewhere between $300 to $450,000 in seed capital to fund immediate R&D and development projects. So to kind of go over why staph is such an important problem for many industries, well, number one, Staph aureus is implicated in many of our medical device infections, things like your IV catheters, hip implants, knee implants, these kinds of things, cardiac pacemakers. Staph is also an important problem to the military. For soldiers that are wounded in the field, biofilms can start forming before they can get adequate medical attention. So that's something that we're interested in developing also, is for military applications to prevent formation on those wounds. Interestingly, staph is also a huge problem for the dairy cattle industry. Bovine mastitis is an infection of the udders of cattle, and these kinds of infections actually cost the dairy industry in the U.S. alone 11% of their annual milk production. That's almost a $2 billion problem faced by the dairy industry. And lastly, there was an earlier talk discussing diabetes and chronic wounds in diabetic patients. And in fact, staphylococcal biofilms contribute in a large part to these chronic wound infections. So overall, in the U.S. alone, we're looking at a $25 billion problem. However, this is also, of course, a global issue as well. To tell you a little bit about our team, first of all, what we've been doing this year is we actually founded our business last October here in Arkansas, and since then we've been working on strategically gathering a advisory board and looking for leaders in the regulatory space to help us strategize how to bring this product forward. My co-founder of PhytoTEK is Mr. Sahil Patel. He has an MBA from Harvard. He also happens to be a dear friend of mine. We've known each other for 16 years and have worked together on a prior venture. Sahil and I recently participated in some of the Harvard alumni business plan competitions and actually came in second in the global finals for our plan. So this has been reviewed by a lot of business leaders, and they think that there's a lot of potential for us to move forward with it. As for myself, I'm an ethnobotanist by training. That means that I study how people use plants in traditional medicine, and I have a very personal commitment to solving this problem of staph infection. I'm actually an amputee. You can take a look at my new leg I got the other day. It's still a work in progress. But when I had my leg amputated at the age of 3, I actually almost died from a severe life-threatening staph infection. I was hospitalized for more than 6 months, and it resulted in a lot of tissue loss and bone loss on my right side. So I'm really driven to look for a solution to multi-drug resistant staph infections, and I really believe that we've found a big part of the solution here in this antibiofilm inhibitor. How did I find this inhibitor? Well, in ethnobotany, I actually travel out into the field. I've worked both in the Peruvian Amazon and throughout the Mediterranean, and the source of this drug is actually from a medicinal plant used in southern Italy to treat skin and soft tissue infection. Those are things like furuncles and abscesses and nasty pussy wounds. So this plant is applied in traditional medicine there to treat these sorts of wounds, and it was through that investigation that I discovered this composition. So what is a biofilm, and how does our extract work to prevent it? Well, when staph enter your body, they do so in this free-floating or planktonic form. So they'll float around your body and they look for a place to latch on and form a stronghold. When they're free-floating, this is the stage at which they're the most vulnerable to attack by both your immune defenses and also by antibiotics that may be given to the patient. So what they like to do is they form a biofilm. An easy way to think about a biofilm is kind of like a force field or a sugary matrix that they produce and hide in. So this actually forms the biofilm actually forms both a physical barrier that antibiotics are not able to penetrate. Again, put in your head that idea of Star Trek and the force field, right? The laser beams can't get through. Same thing that a biofilm does. It prevents those laser beams or your immune defenses or antibiotics from penetrating that secure barrier. The other thing that bacteria do when they're in these biofilms is they actually slow down their growth rate. So most of our antibiotics work on rapidly dividing cells, right? So when they're in this slow growth rate phase and they're protected by this barrier, it's extremely difficult to reach and target those bacteria in this kind of life cycle. So the advantage of a biofilm inhibitor is twofold. The first is that by inhibiting the ability of staph to form the biofilm, you can use it in a prophylactic manner. Some applications of this could be in medical device coatings. Think about your hip implant that's coated with this extract. That would prevent the staph from actually latching on to the device. Another important application is in therapeutics. We found that if you combine our extract with several different classes of antibiotics that work in different ways, you can actually potentiate or improve the activity of antibiotics because you're doing what? You're knocking down that defensive barrier that the bacteria put up, allowing the antibiotics to reach their target. I'm going to show you some of our exciting results. Here you can see an image on the left. This is an image showing the prophylactic testing. On the left, the green and red shows your happy, dividing, growing cells that are attached to a surface. On the right, you can see that this is a sample that was pre-treated with the extract, and the bacteria are not able to adhere to the surface. So it's highly effective in preventing attachment. Most important, though, is in the therapeutic space. When you combine our extract with, again, several different functional classes of antibiotics, you can see up to 100,000 time improvement in the ability of those antibiotics to reach their target and remove the adherent biofilm from the device. So PhytoTEK brings to the table a really unique solution in that we're targeting the biofilm itself. We're reducing the need to carpet bomb patients with excessive antibiotics, again, because we're making those antibiotics more effective. And lastly is what we're preventing that recurrent growth cycle of patients being reinfected and reinfected. What I didn't mention before on one of our earlier slides is thinking back to that idea of a knee or hip replacement. Let's say that you do have a knee replacement that gets infected by a staphylococcal biofilm. At this stage, the only treatment we have out there that's available is to actually go in, remove the knee replacement. So you're surgically removing the device. Surgeons will have to go in and dig out or debride the infected tissue around it. So it's a very invasive procedure. Then you have to be hospitalized under intensive antibiotic therapy. After you've recovered from that, they have to put the knee replacement back in. A new knee replacement. However, the recurrence rate of infection is almost 40% for these. So even after you've gone through all of this, the chances of that knee replacement, the new one, getting reinfected is extremely high. That is because the antibiotics in this procedure are not able to completely clear your system of those biofilm-associated cells. Now, as I mentioned before, biotech and pharma in particular are risk-filled ventures. There's a lot of jokes out there. You have to be kind of crazy or possessed to go after drug discovery, and maybe that's what I am, because I really am dedicated to bringing a solution to the market. We've divided our strategy across both prophylactic and therapeutic applications, and also looking at it based on the pathway to market. As many of you probably know, it costs somewhere between $800 million to $1 billion to bring a new pharmaceutical to market to human use, and about 12 to 15 years to get that done. So to improve our revenue stream coming a little bit closer to us, we're looking at instead first pursuing the veterinary path. In particular, we're going to start with companion animals. So the nice thing about staph, in one sense, is that it affects so many different organisms. So think of it as a broadside to a barn. If you're throwing something at that barn, you're going to hit one of those targets. There's just so many different areas that you could apply this kind of technology to. We're going to start with looking at cat and dog skin infections, and that will cost us around $8 million to bring to market in about 5 years. Next, our next target will be to look at bovine mastitis, again, that huge problem that I mentioned earlier, and then moving forward to chronic wound care and into human applications. Again, we're focusing on a small niche and then working our way up to these larger targets. It's interesting to note that in companion animals, there's about $25 billion a year that's spent on pet care in the U.S. So this is a large market. There is some potential for a good revenue stream coming from that area. Also for bovine mastitis, there's a huge area that could be targeted. And of course, then gradually moving into human therapies. As far as the regulatory stuff goes, we have identified a regulatory specialist who has experience with, she was the lead clinical director for the Merck veterinary area. She's also worked with Pfizer, and she has contacts with Mariel. So I'm presently located in Atlanta, Georgia. I'm on faculty at Emory teaching there, but we also have proximity to Mariel, which is, of course, the major leading animal health care company with products worldwide, products like Frontline and stuff. So that could be one of our potential exit customers for sublicensing our technology for animal wound care applications. Now, our core business can generate between $200 to $300 million in revenue. We believe that we could have a potential of $23 million from the canine soft tissue infection market, 77 from bovine mastitis, and moving up to 169 in chronic wound infection. Again, as I mentioned, there's also a great interest in these kinds of technologies for treating soldiers, so there could be, the U.S. military could also be a potential customer. Today we're seeking $450,000 in seed financing. This will go completely to fund R&D efforts. Our first steps are to hire these renowned regulatory experts to help us map out a detailed path of how to best strategize our testing. Which GLP assays do we need to have done? What are the exact types of drug metabolism studies that need to be done so that we're not wasting any of our money and that we're putting it into the areas that need to be completed in order to get approval through the Division of Veterinary Medicine at FDA. In the long haul, we're looking at needing to raise around $8.5 million to get this first product to market into the veterinary space to treat cat and dog skin and soft tissue infections. And I'll go ahead and stop there. and open up for questions. One thing I neglected to add is our IP status. We've had a provisional patent application in place for the past year, and they're actually filing the utility patent today. I just sent back the papers to the attorney. So thank you.
Thank you very much, Kathy.
Just to make sure, you mentioned about inhibition or suppression. Could you use it for prevention and for removal existing biofilms?
Yeah, so there's applicability in both prophylactic or preventative measures. So an idea there could be as a rinse for these wounds, for example, soldiers that get hit by roadside bombs, to flush out the wounds with this. Another application for preventative measures could be in coating devices, like your central lines or your other kinds of medical implants. In the therapeutic efficacy, the drug is only effective if combined in conjunction with an existing antibiotic. So if you have an established biofilm, it's not able to clear the infection on its own, because remember, this is not a typical antibiotic. It does not kill the bacteria. It only blocks their ability to attach to these surfaces and put up this defensive wall. So when you combine it with an antibiotic, you actually tear down that defensive wall and let the antibiotics work better. Yes.
So as a follow-up to that, from a delivery perspective, does the extract need to be specifically delivered with the antibiotic, or could you take it separately? I'm sort of wondering about FDA regulatory process here.
Yeah, so we believe that it will probably be used, we'll probably pursue first the topical application route in conjunction with existing antibiotics, just because it's easier to prove efficacy when you're treating something rather than preventing it. And one of our first steps will be to go after the active pharmaceutical ingredient process, which is about a $2 million process with the FDA to get that approved, and then we can combine it with other antibiotics. The good thing about that $2 million investment, though, is it can then be used in our other applications. So if we get this API process finished, we can then use that for our human studies down the road. We don't have to repeat that part.
I may have missed it. You didn't talk too much about your source of the antibiotic and the stability of that. I mean, is it plant derived?
It is a plant derived composition. So it's a mixture of ellagic acid glycosylated derivatives, which probably is like mumbo jumbo to many of you, but it's basically a polyphenolic compound. It also has high antioxidant activity. It's found in a lot of your normal food sources. The plant is actually a kind of blackberry bush that grows. So one of the other things that we'll be pursuing is to see if we can get it qualified under GRAS standing by the FDA. GRAS means generally recognized as safe. There is a botanical drug application pathway within the FDA that is specifically has guidance for botanical products that are made up of more than one compound. As for stability, we've done some preliminary stability testing. We're looking at we've identified some marker compounds and looking at HPLC profiles over time. We have not yet completed things like heat stability testing on it as of yet.
Excuse me, I'm more of a business guy than technical,
Yes.
so I may be asking a silly question here, but the process of obtaining the extract, do you do it synthetically, or do you actually have to take the plant and extract it, and if so, how expensive, elaborate, and so on is that process itself?
That's a good question. So from our lab studies have been based on just extraction of the compounds directly from the plant. We believe that this can be done in a good business savvy way, that we can grow these plants. We're also, though, open to other possible synthetic pathways, or also looking at things like hairy root culture to kind of do it in a large scale, or fermentation setup also, are some of the other ideas that we're going to be pursuing.
And so on a $79 treatment, what do you estimate that the cost of the extract to make that treatment will cost you?
I'm not sure at this point. Yeah, that's something we're looking into.
Do you know the active ingredient in the extract?
We know what the large marker compounds are. We know that they are likely these ellagic acid derivatives. It is not, what we found is that when you separate the extract into smaller and smaller pieces, you begin to lose activity. So there may be actually more than one compound that's acting in a synergistic fashion. Now this is a good thing in one way because this makes our IP even more solid, right? And the other good thing is that botanical drug guidance that I mentioned from FDA also takes into account this fact that you can have synergistic interaction of multiple constituents in botanical preparations.
Other questions? Well, we're running a little ahead of time, but we will want to thank all of the speakers for
Thank you.
their participation this morning. Cassie?